Let's first discuss non-adrenergic neurons. As the name suggests, it utilizes norepinephrine as its neurotransmitter. It is synthesized from the precursor amino acid tyrosine, transported into the nervous system from the blood by means of active transport pump. Once inside, it is acted upon by three enzymes in sequence. First, tyrosine hydroxylase. It is the rate-limiting and the most important enzyme in the regulation of any synthesis, which converts tyrosine to dopa. The second enzyme to act is dopa decarboxylase, which converts dopa into dopamine. In the third and final step, dopamine beta hydroxylase converts dopamine into norepinephrine. Norepinephrine action is terminated by two catabolic enzymes. The first is monoamine oxidase A or B, which is located in mitochondria in the presynaptic neurons. And the second one is catechol methyltransferase, which is thought to be located largely outside the presynaptic nerve terminals. The termination of action can also be by a transport pump that removes any from acting in the synapse without destroying it, and is sometimes called the any transporter or net. Once inside the presynaptic nerve terminals, it can either be stored for further use or destroyed by the aforementioned enzymes. It is located on the presynaptic noradrenergic nerve terminals as part of the presynaptic machinery of the neuron, where it acts as a vacuum cleaner. The noradrenergic neuron is regulated by a multiplicity of receptors. The aforementioned net is one type of receptor, as is the vesicular monoamine transporter, that transport any in the cytoplasm of presynaptic neurons into storage vesicles. Now, any receptors are classified as alpha 1, alpha 2 ABC, or as beta 1, 2, 3. All can be postsynaptic, but only alpha 2 receptors can act as presynaptic autoreceptors and regulate norepinephrine release, and are located both on the axon terminal and the cell body near the dendrites. And as we have studied earlier, these are known as somatodendritic alpha 2 receptors. When these presynaptic receptors recognized any, they turn off its further release and act as brake cables for the any neurons, which is also known as negative feedback regulatory signal. Interestingly, drugs that antagonize this receptor will have the effect of cutting the brake cable, thus enhancing release of any. Similarly, the postsynaptic receptors convert their occupancy by norepinephrine into physiological functions, and ultimately into changes in signal transduction and gene expression in the postsynaptic neuron.